For those who don't know me, my name is Moritz, and in the following I will present my thesis with the title Combined Effect of Burrowing Crabs and Tides on Mangrove Carbon Storage that I conducted at the Leibniz Center for Marine Tropical Ecology in Bremen, Germany, under supervision of Professor Martin Zimmer and Professor Niels Moosdorf. Just to give a little background, what are mangroves? Mangrove ecosystems are highly productive ecosystems that are associated with several ecosystem services, such as coastal protection from wind and storms, resources such as food and wood, tourism, and in recent years, their role as blue carbon ecosystems took more prominence. They only account for 0.04% of the global ocean area, but yet contribute up to 4% of global ocean carbon storage. Their capacities to store carbon depends on several biotic and abiotic factors, such as seasonality, mangrove tree species diversity, distinctiveness, primary production, but also organisms that modify the sediment and hydrodynamics caused by tides and waves. Especially many crab species and mangroves sediments act as ecosystem engineers due to their bioturbating behavior and their burrows. They bury leaves into the sediment, preventing the leaves to be flushed out again and preventing competitors to access this limited resource. Therefore, this allows us to hypothesize a positive relationship between burrowing crabs and carbon storage in mangrove ecosystems. However, from a hydrological point of view, depending on the tidal amplitude, this organic carbon might be flushed out again, being for higher permeability of the crab burrows and the processed fragmented leaves. So my thesis aimed at better understanding of how tides and burrowing crabs interact and affect carbon storage in mangrove sediments. Therefore, we tested in an experimental approach the following hypothesis. H1, burrowing crabs increased the carbon content in the sediment. H2, this positive coupling is counteracted by higher tidal amplitudes. For the experiment, we used purchased specimen of Neosamatium africanum, a large semi-terrestrial sursumid crab that occurs in high density in East African mangrove forests, where they play a vital role as engineer species. Quite interestingly, they can bury more leaves into the sediment than the average litter production, which makes them actually an excellent study organism to test our hypothesis of the impact of burrowing crabs on carbon storage, incorporating tidal effects. So as for our experimental design, we worked in microcosms and we used two factors. We used the factor crab and the factor tide. We defined those factors by two levels each. We defined the crab treatments with crabs present, crabs absent, the tide treatments with meso and microtidal elevations. For each treatment, we had 12 replicates, resulting in 48 replicates in total. So on this slide, you can see from left to right our experimental treatments, crabs present, meso and microtidal, crabs absent, meso and microtidal. Our primary source of organic carbon were leaves from the mangrove tree Zoneratia alba that we placed into each experimental unit prior to the experiment. We chose the mesotidal elevations to be around 20 centimeters within the experimental unit and microtidal elevations to be around 2 centimeters. We manipulated our tides using peristaltic pumps that you can see in the middle of this picture. Those pumps pumped for 6 hours from a brackish water reservoir with a constant salinity of 17.5 psu, 6 hours into the microcosms, 6 hours out, resulting in 2 full tidal cycles per day. The experiment ran for 10 days. During those 10 days, we sampled the pore water 3 times, at T0 prior to the experiment, at T1 after 4 days, and at T2 after the experiment after 10 days. We then analyzed the dissolved organic carbon content using high temperature combustion. Also, we measured CO2 fluxes using CO2 sensors using non-dispersive infrared. It's important to notice that the treatments with crabs present, we of course placed the crabs out of the microcosms to assess the microbial respiration and actually not the crab respiration. As mentioned, we had two factors, the factor tide and the factor crab, so 2 degrees of freedom respectively, which leaves us a fully cross-factorial design. We used the response variables DOC and CO2 flux. As for DOC, we calculated the changes of concentration from T0 to T2 and the total outflowing DOC also from T0 to T2, as that differed between the tidal treatments. As we couldn't control for a similar weight among the leaves in the experimental units, we chose POM, so the leaf weight, to be our covariate. This resulted in a statistical approach of a two-way analysis of covariance, testing the effect of crab, tide, and their interaction term on DOC and CO2 flux whilst correcting for POM. We then also had a look into treatments with crabs absent first, where we calculated the DOC leaching rate of the leaves and compared those for tidal effects using a two-way ANOVA approach. Then we had to look into treatments with crabs present and compared the POM consumption, so how much the crabs actually consumed, and compared those for tidal effects as well using a factorial ANCOVA approach. Those leaves from treatments with crabs present, we then also sieved into fraction sizes ranging from 0.5 millimeters to 32 millimeters and compared those for tidal effects using a pair MANOVA approach. As for the results, in graph A, you can see the DOC concentration change from T0 to T2, and on the x-axis, the meso and microtidal treatments. The crab treatments are visualized in colored box plots. Just approaching this graph, we can see that the crab treatments range around similar values for the DOC concentration change. However, in microtidal treatments, the DOC concentration change is higher for both crab treatments. This was then also supported by our ANCOVA with a significant p-value. As for the absolute DOC change, so all the DOC that left the system in graph B, again for meso and microtides and for the crab treatments, visually it seems that there are no differences, and also we didn't find any significant effects of our independent variables. As for the CO2 flux, again on the x-axis, the meso and microtidal treatments and the crab treatments in colored box plots. Just visually approaching this graph again, we can see that meso and microtidal treatments, they range around similar values for the CO2 flux rate. However, when crabs were absent for both tidal treatments, values were higher. This was also statistically significant. So when crabs were absent, there were significantly higher CO2 flux rates. As for the leaching rate, we calculated the leaching rates in treatments with crabs absent for the points in time T1 and T2, which you can see here in colored box plots. In microtidal treatments on the right side of the graph, for both points in time, they were substantially higher, with a p-value lower than 0.01, indicating statistical relevant significance. The POM consumption in treatments with crabs present in graph A in milligram and in graph B just in percentage of initial dry mass, as the leaf weights differed among all experimental units. We can see that in microtidal treatments, they consume more. This was not statistically significant. However, with a p-value of 0.06, we can notice a statistical trend. As for the fraction sizes, in graph A, the dry mass in milligram, again in graph B, in percentage of total dry mass for meso and microtidal fractions, so the fractions on the x-axis ranging from 0.5 to 32 millimeters. Just visually approaching this graph again, in light grayish color you can see mesotidal fractions, and in dark grayish color the microtidal fractions. What we can notice looking at 16 millimeters and 32 millimeters is that mesotidal fractions contributed more to total dry mass, whereas in microtidal fractions, they are more evenly spread for the fraction sizes. This indicates that in microtidal treatments, the crabs, they fragmented and processed the leaves more. This is the differences between those fractions. On your left side are just graphically visualized in an MDS plot showing the differences of the fraction sizes for red mesotidal and blue microtidal treatments. On the right-hand side, you can see the pair MANOVA table, and with a p-value of 0.175, we did not have statistical significance, so the fraction sizes did not differ significantly between meso and microtidal treatments. So I presented quite many results now, just to sum up some of the most important ones. We found a significant impact of tides on DOC concentration and DOC leaching rate with higher values in microtidal treatments. We also found a significant impact of crabs on CO2 flux with higher values with crabs absent. Coming back to our hypothesis, H1, burrowing crabs increase the carbon content in the sediment. We cannot accept this hypothesis as the DOC concentration and the absolute DOC was not significantly impacted by the presence versus absence of crabs. However, the crabs, they buried leaves into the sediment and possibly even increased the DOC content. But we have to notice that they also accumulated leaves into their biomass, which might have counteracted the increased DOC content. Interestingly, the treatments with crabs present had lower CO2 flux values. Why is that? So we measured the CO2 fluxes at the sediment-air interface. And in treatments with crabs present, they buried leaves into the sediment. So there was less carbon sitting on the sediment and on the sediment-air interface where we actually measured the CO2 fluxes. This might have several environmental implications, but also strongly depends on the crab burrow density and stability. As for H2, the positive coupling between burrowing mangrove crabs and carbon content in the sediment is counteracted by higher tidal amplitudes. This we simply cannot accept because we couldn't accept H1. However, we notice a very strong tidal effect. The DOC concentrations were higher in treatments with microtidal conditions. Why is that? In microtidal conditions, the tidal hub was not only smaller, it was also slower. So the leaves were substantially longer underwater, and thus the leaching rates were higher, and thus the DOC concentrations in the sediment. This finding might have actually several environmental implications, as higher DOC concentrations in microtidal conditions, when you compare meso versus microtidal systems, might impact several biotic processes such as nutrient fluxes, microbial activity, composition, and even diversity. Quite interestingly, what we didn't really expect before is that the crabs showed different behavior between the tidal treatments. Even though not statistically significant, they consumed more POM and fragmented the leaves more and overall showed higher activity. When I say they showed higher activity, of course, this is perceived very subjectively. Therefore, we still have some video analysis in process where we placed the crabs underwater and on dry sediment. Why did we do that? Because in microtidal treatments, they were substantially longer underwater, which might have impacted their behavior. Actually, we also noticed this in mesotidal treatments where the crabs showed lower activity during low tide than during high tide. Higher activity of crabs during high tide has been observed before. This might even have implications on organic carbon burial and thus carbon storage when comparing meso and microtidal systems and investigating the effect of crabs to this carbon storage in those systems. Okay. So our study limitations, I just want to quickly mention. We use a very simplified controlled laboratory experiment. So from my point of view, the biggest limitation was the size of the microcosms, because both the vertical and the horizontal movement of the crabs was very limited. We didn't incorporate any interactions between the crabs to prevent pseudo-replications in our statistical analysis. However, when we want to extrapolate our findings to the natural and marine environment, of course, crabs interact with each other, which also might affect the carbon burial of the crabs. Also, with 10 days, our experiment was fairly short, so future experiments might be a bit longer with several sampling dates. As for the outlook, the different behavior of the crabs in the tidal treatments that we found might be a very valuable factor actually to consider in future studies when investigating the effect of tides combined with crabs on carbon storage and mangrove ecosystems. Previous studies investigating the carbon storage from crabs and mangrove sediments didn't really incorporate the feeding of the crabs that in turn might again reduce the carbon burial in the sediment. We have found that tides had an impact on the DOC content, which might affect further biotic processes that also might be further assessed in future studies. So in total, we found very interesting, very unexpected findings. So our study might be viewed as a pilot study, as we were among the first ones, to study the interactive effect of burrowing crabs and tides on mangrove ecosystems. So thanks for your attention. Those are my references, and just very quickly that I'm talking now, I would just like to give thanks to Martin Zimmer and Niels Moosdorf for their tremendous support during my experiments. So yeah, thank you.